Hi everyone, my name is Pham, and I'm a post-bac researcher in the Longacre Lab. And today, I'm happy to present to you our findings on the role of creeping fat-associated fibroblasts in smooth muscle expansion found in Crohn's disease strictures. So before I begin, a little bit of background. Crohn's disease is an inflammatory bowel disease, which is often complicated by the formation of strictures. This scarring leads to the narrowing of the bowel lumen and often requires surgery. One characteristic of these strictures is the expansion of the muscularis propria. However, the cause of this smooth muscle expansion is poorly understood. In our lab, we developed a novel surgical mouse model that replicates this stricture-like phenotype. By creating anti-mesenteric colotomies and closing the bowel under mechanical tension, our model generates strictures with creeping fat and fibrosis. Creeping fat is mesenteric fat that is wrapped around the bowel and typically seen in Crohn's disease strictures. Using this model, we were able to lineage trace MyH11 smooth muscle cells and immunofluorescently stain for ACTA2, a marker of smooth muscle cells and myofibroblasts. On the human side of our analysis, we were able to collect 3 human pediatric samples. We were then able to send these samples for single-cell RNA sequencing and spatial transcriptomic analysis to spatially quantify gene expression and cell signaling patterns. Our results indicate that our model can replicate the smooth muscle expansion found in Crohn's disease, as you can see in Figure 1A, between our sham and our colotomy. Our results also indicate that we have this population of smooth muscle cells that are positive for ACTA2 and negative for MyH11. What's also interesting about this population is that it occurs in the region between the creeping fat and bowel. These results suggest that there are cells here that may be new smooth muscle cells or myofibroblasts. So for our human tissue samples, we partitioned the tissue into 3 distinct regions. These regions being bowel, creeping fat, and interface. We define the interface as the wide area of stroma between the creeping fat and the bowel. Based on our data, we found an increase of gene expression of smooth muscle cells and myofibroblast genes, such as ACTA2 and TAG-LN. This upregulation of gene expression was found in the creeping fat and in the interface. We also saw an increase of Notch signaling, a potent stimulator of smooth muscle cell, in the creeping fat using CellChat. By deconvoluting our single-cell RNA data with our spatial data, we found further evidence that smooth muscle cells and myofibroblasts-like cell populations exist in the interface. From these results, we hypothesized that creeping fat-derived myofibroblasts may be acquiring smooth muscle-like characteristics and contributing to the expansion of smooth muscle in strictures. Our next step in this project is determining if these myofibroblasts and smooth muscle cells originate from the creeping fat itself. Using our mouse model, we plan on lineage tracing these myofibroblasts and smooth muscle cells from the mesentery through local liposome injections to see if there's an infiltration of these cells from the mesentery into the bowel. Furthermore, we will perform single-cell RNA sequencing on these myofibroblasts and smooth muscle cells from the creeping fat to identify different cell populations. Finally, we will culture fibroblasts in the presence of Notch signaling ligands to verify the presence of Notch signaling that we found from our single-cell data. These further pieces of evidence will help us identify if these fibroblasts are truly contributing to the smooth muscle cell expansion that we hypothesize. I hope you learned something today, and thank you so much for your time.